until moving to the University of California, San Diego, where she is the Associate Dean of Global Health Sciences and the Harold Simon Distinguished Professor of Medicine. Her partner in this story and in much of her research is her husband, Dr. Thomas Patterson. Tom is trained as an evolutionary sociobiologist and an experimental psychologist. He's distinguished professor of psychiatry at UC San Diego. And they are both the protagonists of the book about Tom's remarkable story of illness and equally remarkable Steph's tenacity in finding a therapy that would cure a seemingly incurable multi-drug resistant bacterium. So the title today, The Perfect Predator, a scientist's race to save her husband from a deadly superbug. And for those of you who are here, we have copies of the book that Steph and Tom will be available to sign afterwards. And for those of you who aren't here and would like a book, I think we'll have some extra copies in the dean's office. So Steph and Tom, thanks so much for being with us.
Thanks so much, John. It's just a real pleasure for both Tom and I to be here today. And it's a real honor to be presenting in front of Dick Hammond, so with the Hammond Lecture. So as John mentioned, this is something of an unusual kind of presentation because it's very personal. Couldn't get more personal. But first, a couple of disclosures. My husband and I actually were given stock in a phage company that formed as a direct result of his case. We're not going to be talking about specific companies today. I'm also an unpaid scientific advisor to another phage company, and all patient photos are shown with permission, right, honey? Thank you very much. So I'll just start a little bit about who am I and where I come from. When I was a young person, I always wanted to be a scientist, but I was very challenged in math. I actually got a D in calculus, not once, but twice. And when I was interviewing at Johns Hopkins, I made sure that John Samet didn't know that. I also did all my training at the University of Toronto, and my advisor was Dr. Randy Coates, who was a physician epidemiologist who was studying HIV. And unfortunately, he succumbed to HIV himself, and I was on his care team, and it was a very personal loss for me. It was one of the first times that AIDS had hit me in my personal life. But I was able to turn lemons into lemonade because this experience of having lost my PhD advisor allowed me to be recruited to Vancouver, where I became a postdoc before I'd even finished my PhD. And that meant that I had more papers by the time I was an assistant professor than most people had at the associate professor level. And it was just because this terrible thing had happened that allowed me to kind of seize upon it. So I was hoping that I could kind of continue in that tradition. When I decided to be an epidemiology major, my friend's mother said, epidemiology, that's a perfect career choice for you. You've always had terrible skin. Well, double whammy. But I found that it was a perfect match for my skills. And when I left Johns Hopkins after a wonderful time there, I was recruited to UC San Diego, where I worked with Tom, and we started a career together on the Mexico-US border. So this is the Tijuana River Canal because of drought. There's not a lot of water ever running, but when it is, it's always sewage. And that river opening there is where a lot of people actually live in this open sewer. And there are people who inject drugs and trade sex, and that's the work that we have done together, right in the middle of the drug war in Mexico. So we're intrepid travelers, Tom and I. We don't have any problem kind of walking into the lion's den, but we never really thought that we were going to kind of take things as personally as our next challenge took us. This is just a couple of photos from that same Tijuana River Canal. These were taken by the participants in my first study there. And it was a really eye-opening experience. And some of the work that we did attracted international attention because the HIV prevalence in Tijuana was higher than either the US or Mexico overall. And in fact, John and others invited me to present some of the work that I did with Tom at a case study, and it was published in the Annals of Epidemiology alongside several others. And some of this work actually helped my Mexican colleagues as policymakers qualify for funding from the Global Fund to prevent HIV, TB, and malaria to the tune of $76 million. So I really felt like my career was actually making a difference for the first time in my life. And then, you know, something happened that was going to turn everything upside down. But could we really turn lemons into lemonade this time? Well, it really happened when we were on vacation in Egypt. As Tom and I do research together, we travel all over the world, and he wanted to see the Valley of the Kings. It was on his bucket list, and here he is hamming it up. This was Thanksgiving of 2015. You can see that he is much heavier in these photos than he is today, about 100 pounds heavier, to be truthful. And but yet he seemed to be in perfect health. Unfortunately, after a lovely meal, and what was supposed to be the last meal of our trip on the cruise ship, looking over the Nile, thinking we're going to see King Tut's tomb the next day, he got violently ill. And this photo was taken when we got him to a clinic. There was no hospital in Luxor where the ship was based. And, you know, I thought he had food poisoning, but really he just got sicker and sicker. I called home to talk to the head of infectious diseases at UC San Diego at the time, Dr. Chip Schooley, who used to be the chief of infectious diseases here at the University of Colorado in Boulder. And he said, get Tom to like the closest hospital. If there's no hospital, get him to the best care possible, because he needs fluid and probably a whole lot more. Well, he was dead right. And Tom is obviously here in agonizing pain. What they found was something they didn't like. They saw an abscess the size of a small football inside of his abdomen. And it turned out it was from a gallstone that had lodged in his common bile duct. And that wasn't all. When we got him medevaced first to Germany, because he was too sick initially to bring him home. You can see now I'm in full PPE now, which was strange to me at the time. A gown and gloves and a mask, and Tom was fading in and out of consciousness. And the physician came forward and said, we've done an endoscopy, we removed gallstone, and we saw this abscess, and there's this putrid fluid. And he showed me this flask, and it was like brown, murky fluid. And he says, there's something growing in this fluid. If it had just formed this abscess, the fluid would be clear. So he's been clearly walking around with this abscess for quite some time, and something has moved in to make a nice little apartment in there. Well, I wasn't too worried. I thought, modern medicine and this wonderful hospital, they can take care of it. We have these things called antibiotics. But he said, look, we have you in, infection control procedures here in the ICU because we're just taking precautions, because there's a lot of multi-drug resistance in the bacteria that are in the Middle East. So we'll do what we can and culture this. Well, a couple of days later, he came back and he was even in more PPE, right up to his eyes. And he said, look, this is the worst bacteria on the planet. This is Acinetobacter baumannii. In fact, he said, we are almost certain it's multi-drug resistant. We're hoping that it'll be, the garden variety that will maybe be, sensitive to some antibiotics, but we're going to do an antibiogram. So that's essentially an antibiotic susceptibility profile. And whoops, this isn't going forward. What am I missing? Oh, I was clicking on the wrong place. Okay, there we go. So there's the antibiogram. Now, I didn't understand German, and you might not either, but you don't have to be a rocket scientist to see that every R in this chart stands for resistance, and every S stands for sensitive, and it's only partially sensitive to 3 different antibiotics right off the top. And these are what I call in the book gorillicillins, because they're the heavy duty antibiotics like colistin that have major side effects, and they have to be infused into the patient. So now I was starting to get concerned, and Chip had said to me on the phone when I was calling back home, he says, oh, it's not going to be resistant to colistin. The MCR1 gene that confers resistance to colistin was just reported in Lancet this month, and it can't possibly be in Tom. Well, just wait. So they started him on this cocktail of several different antibiotics, hoping that, they would be able to stabilize him. And then we got him medevaced home to San Diego. And so by this time, I'm realizing that my husband is this poster child for this post-antibiotic era that we've been hearing about. That by 2050, it was estimated that maybe superbugs would kill about 10 million people per year. And this would be at a cost to about $100 trillion to the global economy through loss of productivity. These data come from a report from 2016 that was done in the UK. And the data were very weak at the time. They did the best they could. It was the first real estimate. But most recently, just a few weeks ago, the Global Burden of Disease Project put more fine numbers on this and estimated that in 2019, 1.2 million people died directly due to a superbug infection, and about 5 million died with a superbug infection. So we're really already halfway there. So unfortunately, the COVID pandemic has made that worse. And of course, when you put this into a global perspective, it must be realized that antimicrobial resistance will have a bigger burden on lower and middle income countries. In fact, it's estimated that low income countries could lose up to 5% of their GDP, driving up to 28 million people into poverty. So this is something that really had crept up on me. As an infectious disease epidemiologist, you'd think that I would be aware of this, but I really had been focused pretty much on HIV and viral hepatitis. And the more reading I was doing, I was realizing that I had been blindsided by this silent pandemic. And the last thing I'll say about the statistics for now is that, and this was a surprise to me as well, it's misuse and overuse of antibiotics, of course, that's being fueling the superbug crisis, but it's predominantly the misuse of antibiotics in agriculture that's doing this. In the US and in several other countries, 70% of antibiotics are used in agriculture, so in livestock and crops, and even when they're not necessary or it's been shown not to work. And so this is a problem that is obviously going to need a multi-pronged attention. So back to our story. By the time Tom got medevaced back home to San Diego, now this is right before Christmas, so he'd been in the hospital about 3 weeks. The first thing that Dr. Schooley did, because he played a central role in Tom's care, was to repeat that antibiogram that was done in Germany. And unfortunately, even though only a couple of weeks had passed and he'd been in infection control with full PPE, those last 3 partially sensitive antibiotics no longer worked. In fact, this was now a pan-resistant bacterial infection that was even resistant to colistin. And that's what shocked the doctors, because this was the MCR1 gene had just been identified. So how can it be in Tom? And well, it turned out a paper came out a couple months later showing that once the global surveillance had been done to identify this AMR gene, that it was already in 30 countries. So it was no big surprise that it was in Tom. But unfortunately, this meant that Tom was not going to receive surgery to have this cyst removed because there was no antibiotic to cover it. If the organism entered his bloodstream, he would immediately go into septic shock. So what they decided to do was to use interventional radiology to essentially punch holes in his abdomen, put catheters in there to drain off the infected fluid, hoping that his immune system would kick in and that he'd recover. But that was, a very long shot, it turned out. This photo was taken after he'd been in the hospital a couple more months. The infection kept spreading. It's very difficult to get this fluids to siphon off because these drains keep getting clogged. And this organism spread further and further. By the time this photo was taken, he had 4 different catheters and a feeding tube, and he'd lost almost 100 pounds. And the doctor said, look, he's on a ventilator. That means his lungs are failing. He's on 3 different pressors to keep his heart pumping. And now his kidneys are starting to fail. So Steffanie, do you want to start kidney dialysis? And I knew what they were really asking, because that's the trifecta that no patient's family wants to hear. And do you want to extend life support? Your husband is dying. And I decided to have this conversation with Tom, and even though he was in a coma at the time. And I said, honey, I know you're fighting really hard. I want to grow old with you. If you want to live, I need you to fight and give it everything you've got. But if you want to live, please squeeze my hand and I'll try to look into some alternative treatments. And I waited and I waited. And about a minute later, he squeezed my hand really hard. And I was excited, you know, I fist pumped my little gloved hand in the air. And then I thought, oh crap, like, you know, what am I going to do? I'm not a physician. And I have some of the best people caring for Tom right now. But I've got to give it a shot because if he dies, I want to know that I did the very best I could. So what did I do? I went home and I did what anybody would do. I hit the internet. I'm a scientist, so I went to PubMed. You know, our search engine that the National Library of Medicine has bestowed upon us free of charge. And I put in the keywords for Acinetobacter baumannii, multi-drug resistance, alternative treatments. And within an hour, I found this paper that was published a few years ago by researchers in Spain. And it mentioned something called phage therapy. And I knew that phage were short for bacteriophage, which are viruses that have naturally evolved to attack bacteria. I have this rusty old degree in microbiology from the University of Toronto. And I remember learning that what phages were, but I didn't ever know that they'd been used to treat bacterial infections. The more I read, the more excited I got. It turns out that phage had somewhat of an interesting history. As early as 1896, phage activity had been witnessed on the shores of the Ganges and Jamuna rivers in India by Ernest Hankin, a bacteriologist. And he had heard the lore that these rivers were holy and that the water could cure cholera. And he thought it very puzzling that even though this was a site that was very close to a Hindu crematorium where bodies were burned on pyres and half-burned bodies were sometimes tossed into the river, these bodies would go downstream, and the slum that was downstream had lower incidence of cholera than other villages in the region. And so he thought maybe there is something in the water. So what he decided to do was to collect water from this river, and he got as close as he could to some of these bodies. And the paper that he published is pretty remarkable. And he talks about, you know, these buzzards coming down and trying to peck at him, and these, you know, turtles coming up and, you know, trying to eat the bodies. And he wants to get as close as he could to the bodies because he thought that that's where there would be more of a bacterial burden. He collects this water, he puts it through a Pasteur filter, which is going to screen out bacterial particles. And that filtrate he used to put on a Petri dish that had Vibrio cholera on it. And lo and behold, it cleared those colonies. So he said that this water, the Jamuna and Ganges, was cleaner than the Thames, and that the Brits should stop, you know, looking down on the Indians because, you know, maybe, you know, they should be respected. And so it was quite an interesting article. And these experiments, very similar ones, were repeated by researchers in Poland and in Russia. Another Brit named Frederick Twort, who published his paper in 1915 in The Lancet. And he said there's some kind of bacteriolytic agent that is responsible for killing the bacteria, but we don't know what it is. It could be a virus and it could be an enzyme. Well, this was very interesting, and Twort was given quite a lot of credit for this, but it wasn't until a few years later that Félix Decherel, a self-taught French-Canadian microbiologist, repeated the experiment, but deduced that whatever this bacteriolytic agent was, that it must be a virus. Now, only the light microscope existed at this time, so he couldn't prove this, but he said it must be a parasite of the bacteria. And so we're going to name it bacteriophage, derived from the Greek meaning bacteria eater. Well, there's more to this story. There was quite a debate going on, and Félix was quite an egotist, and he ticked off quite a number of the Nobel laureates who felt that Twort was correct and that these were enzymes. And Decherel said, well, whatever it is, I'm going to use this to treat patients with dysentery. And he had miraculous results. These patients were treated and cured within a couple of days. And so Decherel had something of a heyday and brought phage therapy into the 1920s and 30s, where it was used to treat bacterial infections in humans and in animals. And he was the inspiration for the Pulitzer Prize winning book Aerosmith that was published in like 1925 or so. And also at this time, he met this fellow, Georgi Iliava, who's pictured on the right, who was from the Republic of Georgia. And Eliava said, I want to open a phage therapy center and I want you to help me. So de Harel actually helped him do this. And yes, it was the first phage therapy center ever to be erected. And it apparently had Stalin's blessing. Now there comes the rub, because now we're bumping up against World War II. Russia's the enemy. This was known now as Soviet medicine. And that cast a pall over phage therapy for decades. So, and not only that, penicillin had been discovered, and it was brought to market because of the seriousness of the infections on the battlefield. And as a result of all of these factors, phage therapy was relegated to the back burner in the West, and it continued to be used in what is now the former Soviet Union and in Poland, where it's still used to this day. One of the other factors that played a role was several different commissioned reports by what is now the AMA, and a monograph that was published by Gunther Stent from the University of Berkeley, who said that this is a nail in the coffin for phage therapy. He criticized the literature, and when you really look at this paper, you can see that there was real, what we would call implicit bias. He talks about phage therapy being conducted by the enemies in these out of the way places, and that this strange history of medicine this chapter of phage therapy should now be considered closed. So just about everybody in the field read this monograph. Stent was a real luminary, a father of molecular biology. So the last commercial phage preparations were available in the West. They disappeared by the 1970s. So what do we know about phage? Well, some of you in the audience are more experts on this than me, and I was boning up on this while I was trying to figure out whether this could be something we could use to treat Tom. So here's a scanning electron micrograph that was developed in more recent day. When the electron microscope was actually invented in the early 1940s in Germany, phage were one of the first organisms that were visualized, and that's when Felix de Harel's hypothesis was vindicated. You can see here, this is a bacterium stained in orange being attacked by the phages that are stained in green. Now, these are the phages that people see in many different, comics and, on the internet, etc. But we now know that phage come in all shapes and sizes. They're the oldest, most populous organism on the planet. There's thought to be 10 million trillion phages on the planet. That's 10 to the power of 31, or a nonillion. And that's when I started to really get daunted. It's like, oh my gosh, how are you going to find the right phage to kill the right bacteria? Because they're very specific, or finicky, if you will. They have to match to a specific bacterium by attaching to a receptor, and they drill into the bacterial cell, and their genetic material, usually DNA, enters, and then it takes over the bacterium, turning it into a phage manufacturing plant. If it undergoes the lytic cycle, when given the kill signal, the phage, or baby virions, are assembled and they burst don't attack the bacteria that don't have the receptors. So, so if you're using phage therapy, it only is going to kill the bacteria that they're a match to, and not harm the other bacteria in our microbiome. Now, contrast that to antibiotics that are used often broad spectrum. Those kill the friendly bacteria in our microbiome, and we know now that that's not a good thing. So we know now that there's lots and lots of different kinds of phages, but phage therapy is still in its infancy. And then my next question was, well, if I was going to try to use phage therapy to cure Tom, where would we get it from? Well, it turns out that some of the best place to source phage is where you find a lot of bacteria. Turns out that would be sewage, bogs, barnyard waste, wildlife scat, like some of the most horrible places that you can imagine. So here's a flask of sewage. And the next slide shows, let's see, here is a Petri plate streaked with Acinetobacter baumannii. Those globs are bacterial colonies. And if you want to see if you have phage that will kill that bacteria, so you put a drop of, say, sewage on this plate and you incubate it for 24 to 48 hours. And if it comes back looking like Swiss cheese, you see those little holes, even though the phage are 100 times smaller than bacteria, you can't see them with your naked eye. You can see that they've been at work. They've gobbled up those bacterial colonies. So you get excited. You think, okay, I've got a phage that's going to kill the bacteria that I want to kill. And you pluck it out with, say, with a pipette. This can all be multiplexed to make it much more efficient, but this is the simple plaque assay, as it's called. And then you can add more bacterial suspension and grow that phage up. And then you need to purify it before you're going to treat the patient. So I got really excited now. I'm thinking, okay, can we use phage therapy to cure Tom? And the first thing I did was write Chip Schooley an email to say, okay, I understand that this phage therapy is considered a little fringe or woo-woo, but do you think we could try it to cure Tom? And he wrote right back and said, what an interesting and intriguing idea. If you could find phage to match Tom's bacterial isolate, I'll call the FDA and see if I can get permission to use it on a compassionate basis. Well, I was extremely excited that I had somebody who was going to partner with me on this, what seemed like a crazy idea, in injecting extract from sewage into your husband to kill his bacterial infection. But he also was telling me something that he hadn't admitted, that Tom was dying. His compassionate use means the patient has no other recourse. So he said, go see if you can find some phage. Well, a student had given us this t-shirt, I survived Iraqibacter. It's the unfortunate nickname for this Acinetobacter baumannii because so many veterans had come back from the Middle East with this infection and succumbed to it. And I went back to the internet, made a list of all the people that were studying phage and Acinetobacter baumannii, and I wrote them cold, begging for their help. And luckily, within 24 hours, I heard back from a total stranger, Dr. Ryland Young at Texas A&M, who said, your story really struck a chord with me. I'm the same age as your husband. I've been studying phage my whole life. I'd really love to see phage therapy come back to the West, and maybe you're somebody who could try to make that happen. So if you can send me his bacterial isolate, I'll turn my lab into a command center, and we'll look at the phages that are already available to us, as well as our environmental samples. And he says, you know what I'm talking about, don't you? And I went, yeah, sewage. He goes, yep. And so he got all of these folks on the right-hand side of this slide to dump everything that they were working on for the next several weeks to months. And the young woman with the long necklace was a PhD student working in the laboratory who slept in the lab looking for phage to match to Tom's isolate. And she found 4. And so I told Chip, he said, that's remarkable. I'm going to call the FDA right away. And not only that, Dr. Young reached out across the pond to try to find other phages as well, because the thought was that we want as many different phages as possible to try to ensure that the bacterial isolate wasn't going to turn into a mutant that would evade the phage. And one of them was Dr. Jean-Paul Pirnay from the Royal Astrid Military Hospital in Brussels. And he offered to send his phage to us in a diplomatic pouch. Well, this is important because when Chip called the FDA to see if, they were going to be supportive of this idea, not only were they supportive, they knew all about phage therapy. They were looking for a case like this, a patient who was dying, a physician who was willing to try it, a phage community who was willing to source phage for it, and a family that was willing to take the risk. And so when the Royal Astrid Military Hospital said that they would send phage, that convinced a colleague of ours who we learned was doing research from the military hospital. Actually, this is the US Navy Biological Defense Research Director at the same unit that had worked on the anthrax letters from after 2001. And this is Lieutenant Commander Theron Hamilton. He was approached by the FDA to say, look, we know you're working on phage. Would you talk to Dr. Schooley to see if, you might be willing to help him with a patient? So the FDA, has this reputation of being a part of this, but he was put to work and he found 4 phages that were a match. So that was pretty remarkable. And now, unfortunately, Lieutenant Commander Hamilton had to go and tell the admirals, we've got this case. We have phages that could cure this guy. Can I send them to UCSD? And the admirals went nuts because he hadn't asked to do this first. And this part isn't in the book because the Navy wouldn't allow me to put it in. But apparently one of the retired captains who was an MD said, look, admirals, we have these phage that will cure this man's infection. And not only will this jumpstart the field at least 10 years, we will beat the Army. And that's what did it. So he said, hit the go button. And so now we had 2 phage cocktails. Now, in the meantime, Dr. Schooley was doing his homework because he's never done anything like this before. And he had to figure out things like for the protocol, like how much phage do you administer when the drug is alive? What routes of administration? How often do you treat the patient and for how long? And he consulted with Dr. Maya Mirabishvili, who'd worked at the Ilyava Center, which as it's known is still in the Republic of Georgia, as well as Dr. Carl Merrill, who was a phage researcher at the NIH, whose lab was actually closed down by Tony Fauci when he said this isn't going to cure AIDS and it's like a pipe dream. And Carl said, look, Chip, you need to treat this patient intravenously because if there's a hidden reservoir of bacteria that the phage don't reach, it can become resistant right away and you don't have any other phage left. So Chip agreed and he mentioned this to me and I knew that it was coming because I'd been doing my homework. And I said, yeah, we got to do it. So here's the phage cocktail that was ready within 3 weeks of my first email to Rye Young and others. And half of the time was spent scrubbing the endotoxin from this preparation to make sure that it wasn't going to be something that was going to cause septic shock in Tom, because he'd already had about 5 cases of septic shock by this time, and he wasn't going to live through another, nobody thought. And it was approved by the FDA through what's known as an EIND, or an emergency investigational new drug. And the dose that we arrived upon was pretty much a bold guess, 10 to the 9 PFU per mil every 2 hours. And that's essentially 1 billion phage per dose. So you can imagine that we were pretty terrified when this happened. This is what Tom looked like on March 15th, the day that we started phage therapy. He looked like a pincushion. He was hanging on by a thread. I had to sign the consent form for kidney dialysis the day that we started, but we never needed it. This is Chip smiling for the camera with our colleague, Dr. Randy Toplitz. They knew that they were making medical history, whether the patient lived or died, because this turns out it was the first time that a patient in the US had been treated intravenously for a systemic multi-drug resistant infection. And so we started with the phage that came from Texas, and those were first inserted into the catheters in his abdomen because that was the closest to the source of the infection. And the thought was, if he lives through this, we're going to get the Navy phages, which were more virulent, and we'll put those into his PICC line and treat him intravenously. So that's what happened. And on the right-hand side, I stood there and watched these phages infused into his body. And, Tom's daughter, Jessie, who's going to be joining us at the reception, turned to Chip and said, well, what happens now? And Chip says, we wait and we hope that we have the most boring 24 hours that we've ever had because we don't want septic shock. Well, 3 days later, Tom lifted his head off the pillow, opened his eyes and kissed his daughter's hand. And nobody thought that he was going to recover from this. I learned later that they thought that he was within hours of dying. And the pulmonologist that was overseeing the case at the time said, look, this is like a Hail Mary pass at the last minute of the football game where the quarterback is blindfolded and he's just throwing the ball 100 yards down and hoping that somebody will catch it. And by God, somebody did. So Tom's recovery was very tumultuous because he had lost so much weight and all of his muscle mass. He had to learn to stand, walk, swallow, talk, everything. And he finally left the hospital 9 months after his ordeal began. And a few months later, he actually met Lieutenant Commander Theron Hamilton, who came to visit us. And you can see that Tom is looking much better there. He's still 100 pounds lighter to this day, but it's all good. And the case report was published by Dr. Schooley in a good journal, AAC, but it was actually submitted to the New England Journal, JAMA, and Lancet, all who passed on it. The editor of NEJM apparently wrote back, said, I don't believe it. And later we were told that, this historical geopolitical bias that had hung over the field for the last couple of decades had really played a role in their decision, because all of those journals kind of changed their mind later. I'll tell you about that. So one more electron micrograph is that here is Tom's bacterial isolate that is stained in blue being attacked by the navy phages that are stained in green. And this scanning electron micrograph is it's magnified 100,000 times, and it was courtesy of the National Biodefense Analysis and Countermeasure Center of the Department of Homeland Security. Just getting this photo into the book, because it's in the middle, took like months of approvals, but it was worth it. So after Tom's case was presented at the 100th anniversary of the discovery of the bacteriophage at the Pasteur Institute in Paris by the phage whisperer, Dr. Biswas, he got a standing ovation, and the story went viral, but in a good way. So patients started calling me and Chip from all over the world saying, could we get phage therapy for my family member or for my patient? And because total strangers had stepped up to save my husband's life, I felt like we needed to do the same. And so we reinvented the wheel. We did the same thing. Sometimes we saved the patient. Sometimes we couldn't find phage in time. And then the press started to pick this up. So we were in, Huffington Post. We were in the Washington Post. We were in People Magazine. We were on the Today Show. Time Magazine, named me as one of the most influential people in healthcare, the top 50 in 2018. And like, healthcare? I'm not even a doctor. So that was pretty amazing. And then these journals that had passed on the case report, JAMA published a Q&A with Chip Schooley. The Lancet commissioned a commentary on phage therapy, mentioned Tom's case, and also published a book report, a book review rather, by Laurie Garrett, who's an eminent scientist, communicator, and had published a lot of books like The Coming Plague. So, it was really kind of ironic. But then we started to treat more patients. And that is what led our chancellor at UC San Diego to say, this is actually the most impressive story I've heard since I came to this university over 6 years ago. He said, I'm going to give you seed funding to start the first dedicated phage therapy center in North America. And we called it the Center for Innovative Phage Applications and Therapeutics. Chip and I are the co-directors. When we opened our doors in June of 2018, we had a commentary in Science where an eminent microbiologist said that this is a game changer for the field. We've treated many other patients since. We don't even know how many requests have come into the FDA because they don't allow us to know that number. This is just a smattering. Tom was patient number one, of course. We've treated patients with different kinds of superbugs like Pseudomonas aeruginosa, ESBL E. coli, and others, and different underlying health problems like idiopathic pulmonary fibrosis that leads to many different infections, cystic fibrosis patients that get chronic lung infections, people with implanted devices like LVADs or prosthetic joint implants. We've had some success. Sometimes we clear the infection, but the patient would die of their underlying disease because these were all compassionate use cases. And we published these as either case series or case reports, and the field has been taking off internationally. There's many other phage therapy programs now. The Brussels program has a formal program. Baylor has a program. Yale has a phage therapy program. The Mayo Clinic in Minnesota has a phage therapy program that specializes with prosthetic joint infections. And when you look, though, at the requests that we get into IPATH, it's not all rosy. And that's because the majority of the patients that want phage therapy aren't eligible for it because it's still an experimental treatment. If they have an infection that's susceptible to antibiotics, then the FDA won't approve it on a compassionate basis. So it has to be a fully resistant infection. So not indicated, or 855 people who wanted phage therapy that were turned away. Now, when phage therapy has been recommended, we have to go on a phage hunt. We have to find phage that are going to match the bacterial isolate. Sometimes we get lucky, and these are phage that were identified off the shelf and that have been sitting in a phage library. Other times we have to go directly to sewage and bogs and things like that. And then sometimes the patient dies before we can get the phage ready for them. And sometimes we can only find these sleepy phages or temperate phages, as they're called, and those do not all these little phage libraries all around the world, and it's like having a million locks scattered all over the place and having a million keys, and you have to find the keys that match the lock. So our goal is to build a phage library that will cover the majority of escape pathogens that are of biggest threat to human health. In some cases, we might be able to have fixed phage cocktails, like for staph. 20 to 30 phages are thought to cover about 80% of the circulating staph isolates around the world. But for something like Acinetobacter baumannii, the bacteria that was killing Tom, you have to match it to the isolate, not just the genus and the species. So that becomes more challenging because you may need 300 phages or so, but we don't really know. We need to use microbial ecology, population dynamics to, inform this. And then we have other kinds of bugs like mycobacteria, and I'll talk about that in a second. Chip and I have done some publishing. I'm more on the advocacy side, where Chip is involved in the clinical trials. We published a paper in Nature Microbiology, treat phage like living antibiotics. It was really our roadmap to say, don't rush into phase 3 efficacy trials before you've done the translational studies that are going to inform them, because this field has had enough of a cloud over it. We want to do the research right. Now, luckily, the NIH has bought into this. When we have someone who's as credible as Chip Schooley giving presentations like ours, people believe it. And so the NIH funded their first phage therapy trial to the tune of $12 million through the Antimicrobial Resistance Leadership Group, which is a research network across the US that has been funded to test out new antibiotics. But as we know, there's not many new antibiotics in the pipeline, which is drawing up. So they prioritized phage therapy. Now, this was actually funded right before the COVID pandemic began. So it's been put on hold, but we're just about to start enrolling our first patients. And Chip is one of the PIs, along with Dr. Tama from Johns Hopkins. So we're very excited about this. In fact, when you look at clinicaltrials.gov, which is a registry of all NIH-funded clinical trials, and you look specifically at phage therapy trials, you can see that there's been an increase. So we're not the only ones doing phage therapy trials, and that's good, because we need lots of different trials testing different kinds of organisms and different kinds of therapeutic doses to be able to inform FDA decision-making around the future of phage therapy. Now, I have one more case to tell you about, and this is with Mycobacterium. I mentioned that they're tricky, and this is because with Mycobacterium, like several other bacteria, the only phages that we can tend to find are temperate phages, those sleepy phages that enter the bacterial cell, integrate their DNA into the bacterial cell DNA, and hit the snooze button, as I describe it in the book. Now, we don't want to use those phages because they don't kill the bacterial cell, and they can sometimes maybe potentially cause harm. There's some debate about that. So what was done in this case is that this is a CF patient who'd had a double lung transplant and had a disseminated infection of Mycobacterium abscessus, which is a cousin to tuberculosis, the biggest bacterial killer in the world that kills almost 2 million people per year. Now, we were approached to try to help find phages for Isabel. And we learned along the way that there's this wonderful training program called CPhages, and CPhages actually exists here at the University of Colorado. I met some of the students this morning, maybe they're online watching. And the CPhages program is funded by HHMI and founded at the University of Pittsburgh. And there's over 200 different institutions around the world that offer it. It's a training program for undergraduates, teaching them about virology. And they, do essentially what I showed you, the plaque assay. And if these students find a new phage, they get to name it. They get to sequence it, they get to annotate it, and it goes into a phage library. But nobody ever dreamed that those phages could have therapeutic potential. And when we, realized that there was like over 10,000 Mycobacterium phages in this library that were sitting there, somebody said, we've got to see if these can be a match for Isabel. And Dr. Hatfull at the University of Pittsburgh said, absolutely, he would look. And he had his lab, do a very intensive search, and he found one lytic phage that was a match. And it came from a rotting eggplant, or aubergine for your provost, that was sourced from a rotting eggplant in South Africa by a student there. Okay. And so one phage is not going to be enough. You want more, but he could only find 2 temperate phages. So what he did was a predecessor to CRISPR-Cas gene editing is called recombinering. And he used recombinering to clip out the repressor gene that makes those phage sleepy. And he forced those phage to become lytic. So now essentially we had the first genetically modified phage cocktail. Now the next trick was, are we going to be able to convince the UK government that this is not a GMO and that we could use this cocktail to save this dying girl? Well, there was a debate about that, but ultimately they said, well, you know, a gene was taken away and a gene wasn't added, so it's not a GMO. So yes, you can go ahead. And so Isabel was in hospice when she received phage therapy. The doctors were initially going to sprinkle the phage on her skin, and Chip says, absolutely don't do that. She's fully colonized, just like Tom. Here's the protocol, go intravenous. And her mother said, yeah, let's go for it. She's got a 1% chance of living. Let's do it. She left the hospital within a week. That's how quickly phage therapy worked. And she had an extra 3.5 years of life. She eventually just passed away about a month ago. And we had become very close because we befriended each other on Facebook. So her case has really been remarkable because it's made biotech and pharma realize, wow, you know, we could patent much easily more than natural phage, a genetically modified or a synthetic phage. So there's a lot of new research being done in this area. And it also shows the power of citizen scientists like undergraduates, that they can source phages that can be used to treat somebody. And that's pretty powerful. So I'm going to end in a minute, but I did want to make sure that you were aware that the COVID pandemic has made the AMR crisis worse. There's been a number of reports that have shown that there's been a significant increase in central line associated bloodstream infections that are associated with sepsis, catheter associated urinary tract infections. Anybody on a ventilator is much more prone to infection. And so we've got our work cut out for us. Myself and Dame Sally Davies, who's the AMR envoy for the UK, published a paper in Lancet in 2020 talking about how COVID is really an opportunity to be able to get ahead of the AMR crisis as much as we can. And we propose different strategies for doing so, including embracing a One Health approach, which is really a global health paradigm that considers the interface of animals, humans, and the environment. We need a multi-pronged approach if we're going to tackle AMR. Phage therapy is just one of the weapons in our quiver. So there are a number of other phage applications that I don't have time to talk about today, but I want to make you aware that they are being examined to replace antibiotics in livestock and agriculture and even aquaculture. They're being examined for food safety. In fact, there are some phage preparations that already exist to decontaminate meat against listeria or salmonella. They've been used to prevent foul brood in apiaries and to be used as prophylaxis during cholera outbreaks. Phage are being studied to groom the microbiome to weed out unfriendly bacteria in our guts that might cause harm. And phage are even now being used as a vector to deliver cancer therapeutics or vaccines, because from Tom's case, we know that injecting 1 billion phages every 2 hours into somebody who's very sick is not associated with any side effects. In fact, we haven't seen any side effects from intravenous phage therapy in any of our patients. There's also now interest in using phage to disinfect wastewater and to remove sludge in pipes in the middle of the ocean. And then finally, unfortunately, we're living in a dangerous world right now where there are, bacteria that are used as bioweapons. And so it's no coincidence that the Navy and the Army have been working on this, because phage could be seen as to counterattack against an anthrax attack. God help us. So with that, I think our story tells you that when life, gives you lemons, you can make lemonade. But in our case, we made a phage cocktail. So with that, I'll just acknowledge that there's many, many collaborators that we have worked with and continue to work with. And we'd love to see the University of Colorado's phage hunting program help source phage for patients if they're interested. A global village stepped up to save my husband's life. We haven't even met everybody. It really puts the kind in humankind. And, my view on this is that miracles don't happen, they're made. And with that, we realized that we are privileged. We, had wonderful people that came to our rescue and connections that other people don't have, which is why we wrote our book, The Perfect Predator. And it's done well. We've had Hollywood knocking, and we're using this as a platform to open the discussion about AMR and to ensure that this 100-year-old forgotten cure gets its fair shake. So with that, I'll close and see if there's time for any questions. And Tom will join us too. So thank you.
Let's see, Tom, do you want to join in? What I'm going to suggest, if I can figure out how to see the chat, that we we'll start there, and then since we're a small group here and there's stuff to eat in the back, we can probably just gather and have a conversation back there. So let me see what sits in chat. Problems with AV. Let's see. Sound is off again. Thanks for help. Sorry. Okay, well, hopefully those of you, I hope everybody heard this, and if not, we've got a lesson learned, but let's see. So I don't see anything in chat. Oh, here, let's see. How do you do a randomized trial with phage therapy? It comes from Lori Crane. So you want to take a crack at that?
Well, it's tricky because the drug is alive, right? You know how much phage you're administering to the patient, but you don't really know how much phage the patient is getting because the phage will multiply as long as they have a bacterial target. So it's tricky in that way. But, at this point where we're starting with the phase 2 trial that Dr. Schooley and Dr. Gemma are leading is that we're working with patients that are not receiving antibiotics. So these are chronically infected patients that don't have a fulminant infection that requires antibiotics so that we can eliminate the confounding effect of antibiotics when we're seeing how phage are working. Then we're going to do pharmacokinetic and pharmacodynamic studies to try to, pinpoint what the adequate dose will be and the routes of administration. From that, those kind of bedside to bench studies, or as I refer to them as bog to bedside studies, will inform how to go about an efficacy study.
Before we leave the remote platform, Tom, I want to see if you want to chime in. I mean, Tom has remarkable things to say about his experience, and if we could take the next hour, we would begin to get a tiny insight. So please, why don't you?
I'll just say a couple of things. First, I'll just acknowledge how privileged I was to first chose so wisely to pick a wife who was good at mobilizing the world and coming up with this idea, but also just, the family, the physicians, nurses, and all of the researchers that really chipped into this. One major lesson that I always like to point out is, though I was in a coma for 3 months, I wasn't a loaf of bread. I could hear not every single thing that people said, but the message to you as a provider, perhaps, or a family member that's visiting someone, be careful what you say because it'll affect how people are thinking. My hallucinations were driven a lot by conversations and physicians talking about futility, etc. So that would be a big point for me to point out for you. I think I'll stop there.
Okay, well, I think what we'll do, and let's see, here's a couple more questions. Impact on the microbiome, and how long does it take to get a match? And I guess that's really the problem you're working on, so that will be as short as possible.
So the impact of phage on the microbiome, well, I can certainly say it's less than antibiotics, but we still have to have a better understanding because we know that phage are specific to certain bacteria. And if the bacteria doesn't have the receptor that the phage matches to, then it can't affect that bacteria. But there are prophages. Those are the phage that have integrated into bacterial cell DNA and inside of us all the time. In fact, there's been an estimate that 30 billion phages move in and out of our tissues every single day. So they're the gatekeepers in our microbiome. They're already part of us. It's just that they're what's called a viral dark matter. We haven't really had the techniques to culture them and study them before metagenomics and high throughput sequencing. So we need to have a better understanding of the interface between phage, bacteria, and the immune system to be able to really pinpoint how to move forward with phage therapy. But at this point, we haven't seen any untoward side effects of phage therapy. In terms of how long it takes to match phage, it's highly variable. If we get lucky and we have phage, like if somebody contacts me today and they say that they have a Klebsiella pneumonia infection, I know exactly where to go. I know that Baylor has some really great Klebsiella phages, and I know that Yale has some, and our lab at IPATH does. So I can say, well, if you're willing to send the patient's bacterial isolate to those 3 labs, and those 3 labs aren't overwhelmed with like COVID right now, and they agree to do a phage hunt, they could say, well, we've already got phages in the library for that. And if we have a hit, we could have a phage cocktail within a couple of days. And then we have to, get those phage prepared and then sent back to wherever the patient is. And then we help the physician, put in the request to the FDA and put the protocol together. And we have that physician learn how to administer phage therapy. We don't ask the patients to come to us. So it can be just a couple of days, but sometimes, especially when we have these temperate phages, it can take months. And that's why we need a phage library. So what I'm trying to build now is a private-public partnership to scale up a giant phage library that could exist on every continent. And that's kind of my vision. And the other part of the vision is to take the C-phages program to the developing world and to have citizen scientists in lower and middle income countries learning, but also sourcing phage from those environments to add to the phage library. And that'll be a win-win for everybody.
Great. So how many facilities, that's maybe the last question, we've gotten there. How many facilities actually do this now?
Well, the programs in Poland and the Republic of Georgia are for-profit. They've been operating for a long time, but Brussels has a phage therapy program. I've mentioned Baylor, Yale, the Mayo Clinic. They're more on the clinical side, and then ours. So there's programs that are trying to get going in Canada as well, but there's a lot of biotechs and small phage startups. Many of them are involved in phage hunts as well, because if they can help identify phage, then their stockholders get happy. So we reach out to them. The Navy does not involve itself right now with phage preparations for patients, but Walter Reed has stepped up to the plate. So the Navy moved on and the Army moved in.
So I want to thank you both so much for extraordinary presentation, and what a story. And, I think you are really trying to democratize phage therapy, basically. And I think, so it's probably, hopefully not a question of how many facilities, but can everybody have access to one when the time comes? And I guess hopefully the libraries will be at the ready. And that is, given what's happening with microbial resistance, we need this. So again, thanks so much. A great second Hammond lecture, and you're coming back sometime. Thanks. Thanks everybody online, and sorry for our delays.
We're available to sign books if anybody would
Yeah,
like.
so come on to the back.